Good afternoon. This is Chapter 1, A View of Life, and essentially what it is the author's view on how one should think about the study of biology. I'm gonna get rid of my face down there. I mean, some of you may be crying, but you're gonna just have to live without it. I'm just gonna do voice for this one so that we can see the whole slide. So here we go. Chapter 1, A View of Life. We're going to start with the scientific method, which actually comes towards the end of the chapter. Make sure you pay attention to the reading assignments. You don't have to read quite all of the chapter. I tried to cut down on that for you. So this is towards the end. The scientific method. And we can just start with a smiley face there. Right. So I'll read through them real quick, and then I'll describe this experiment over here that is also in your book. The scientific method involves first making some observations, then you form a research question. Typically, it should be, it almost never is, but for instructional purposes, it's good to think of a good research question as a yes or no question, such as, does this medicine cure this disease? Answer is yes or no. And then researchers will develop a hypothesis, which is, for now, we'll just say basically kind of a prediction about something. Independent variable and dependent variable. Speaking of hypotheses, a hypothesis basically is how does some independent variable affect a dependent variable. And we'll look at quite a few examples momentarily. Then there's these controlled variables as well. And that is basically everything else in the world that you try to keep the same. There's an experimental control group, sometimes also called the control group. So these two things are the same thing. I'll give examples. It'll become clear what those are. And then also, good research or good science must be repeatable. It should be free from bias, and it should be randomized. So these are some things we're going to be thinking about as we go through a couple of examples. Let me think. Let me just give you one example of bias right now. Bias would be, for example, if a company such as ExxonMobil did funded research looking at, say, the burning of fossil fuels and their effects on the environment, then that would obviously be biased because ExxonMobil sells fossil fuels, and so they obviously have an interest in the answer to that research question. The research question being, does the burning of fossil fuels harm the environment? They want the answer to be no, so that would obviously be a problem. There would be bias there. All right then, so let's take a look at our first example here, and let me direct your attention right up here. This is where we have our research question, and it is the nucleus essential for the well-being of the cell? So here's the nucleus right here, and then this thing is the cell, and basically somebody came up with this research question a little after the first microscopes were invented, and they basically saw that inside pretty much all cells, if you looked enough, you would find some kind of blob in there. Eventually they came up with the name nucleus, and that was often all that they could see inside of a cell. So the question was, is that thing we can see in there, is it important? So what they did was they thought, well, let's remove that thing in the cell, the nucleus, and then see what happens. And what happens? We got this hook. Somebody's really good with tiny hooks, and they stuck this tiny hook into the cell, and they pulled out the nucleus. Okay, it's pulled out now. No nucleus in this cell. And what happens? Well, this cell dies. This is an amoeba. They're pretty cool if you ever get to see one. The amoeba or the cell has died. And so they might be tempted to conclude right away that if you take out the cell, then if you take out the nucleus, then the cell is going to die. Problem with that conclusion is as follows. It may have been that simply by collecting these cells, I mean, cells don't just jump into your lab and ask you to experiment on them. It may have been that something about how you collected them was in fact what killed them. It just so happened that you also took out their nucleus before they died, even though they were already on the way to dying. Another thing is this: that the experiment itself could have killed this cell. Sticking hooks into living things is generally a bad thing for the living thing. So what we have to do is have two groups, group number 1 and group number 2 down here. And the group I just described, that's the experimental group. We're going to give it the treatment being removal of the nucleus. Down here is the control group, and I wrote that over here as well, control group. We could also say it is the experimental control or the control group. Both of those would be accurate, and make sure that that you know the difference between controlled variables and these two things, experimental control and control group. We'll come back to that. Easy to get those mixed up. So what is the use of group 2? Basically, the experimental control serves as a comparison. What we want to do is have these cells, and there would be dozens or hundreds depending on how much time the researcher has, but this group having lots of cells and this group, they are treated in exactly the same way except for the independent variable. And the independent variable in this experiment is removal of the nucleus, or you could say it a different way, has or not a nucleus. Does it have a nucleus or does it not have one? So everything else is the same. Here, what they do is they'll even go to the time and expense of jabbing a hook into the cell, maybe poking the nucleus, and then pulling out the hook, but leaving the nucleus here. So both groups, the experimental group and the control group, experienced all of the same things except the independent variable. The independent variable being removal of the nucleus. So down here, these fellows survived. The control group survived. We can conclude that the nucleus is important to the well-being of the cell. And the well-being of the cell is not the greatest research question. It should be a little clearer, such as is it necessary for life? Well-being is not something that you can measure. You can measure dead and alive. That can be measured. Okay, so let me just talk really briefly about controlled variables. Controlled variables would be all the other things that you are trying to keep the same in all treatments of every single cell, such as easy one students will always go for if they're asked to name a couple would be cells in the experimental group and the control group must be kept at the same temperature. They must be fed the same type of food or medium if we want to be more sciencey. They must be cells of the same age, cells that we found in the same location. For example, you couldn't have found the group 1 or the experimental group. You couldn't have found these cells in a polluted gutter and then gone to a pristine pond and gotten these cells and then done your experiment. That would have some obvious problems because you failed to keep some of the controlled variables the same. Okay. Let's see here. So let me just touch on these last two here. Repeatable, yes, the experiment's repeatable, so that's great. If you designed an experiment that was so amazing that only you were able to do it, there would be some problems. People would not believe you. And then randomized. What does that mean? Well, it would mean you go down to the pond, you get yourself maybe 150 cells, and then you randomly select, say, you flip a coin as you pull out a cell from your pond sample. You flip a coin, and if it's heads, you put that cell in the experimental group. If it's tails, you put that cell in the control group. So you're going to randomize how each cell is put into each group. All right. Here's another example of the scientific method. The village cure. I'm going to read through this one with you. In an imaginary village where people have been living for thousands of years, much is known about the local flora and fauna, and this knowledge is passed down from generation to generation in the written form and also by the oral tradition, meaning talking. When a person in the imaginary village gets a cold, they boil the leaves of a local plant, which produces something we will call the cold cure. The sick person drinks the cold cure and then lies in bed until they feel better. People in the village claim they have a cure for the cold. Discuss. What value does their claim have? Is this science? Is their claim justified? You can pause the video here for a moment and think about it. Then I will discuss it. So pause it or not. Okay, if you pause it, well, I'll just assume you have. So moving on. What value does their claim have? Well, firstly, absolutely, they have been making observations for a long period of time. They've obviously been wondering, do things cure the cold? Does this leaf cure the cold? They have been doing an experiment of sorts where they give somebody who is sick a treatment and then wait to see if they get better. So there is value in this, and it has aspects of the scientific method. However, there are problems. Firstly, there is no experimental group. Uh, in this example, in this village, they didn't have maybe 50 sick people and then randomly assign each of them to the experimental group and then the other ones to the control group. So they don't have anything to compare the people who got better to. They would need to give an experimental control group a placebo. That would be the best way to control all the variables. Each person believes that they are getting the cure, and they don't know if they are getting a fake cure or a real cure. If you've ever heard of a double-blind study, that is when researchers give doctors a cure for some disease, and sometimes they're giving the doctor a placebo. That means a basically saline solution or just some kind of liquid or pill that actually has nothing in it, like a sugar pill. And then the doctor themselves, who is treating the person, they don't even know whether or not it is the real cure. The reason that they would do that to make a really effective experimental control group is that there's something called the placebo effect, and that is that when people are given pretty much any treatment, there are measurable benefits to their body. Their blood pressure may go down, their their pain may be reduced somewhat, and that is simply that the stress response is toned down a little bit, and the parasympathetic nervous system's activated, and they just feel a little bit better. So everything has got to be controlled. There's got to be an experimental control group. All right, going a little long on this one. So let me wrap it up. Their claim isn't justified, unfortunately. Just one other thing with disease. The problem with diseases is that often, especially with the cold, people just get better even if they do nothing, especially if they are lying in bed. All right, so this does have value though, and that is simply that they have been making observations for a long time, so it does have value. And in fact, many traditional medicines, such as those in India, really old culture, they've been making observations and writing them down for thousands of years. China as well. Modern researchers are going into that library of information and looking for, well, basically plants that may have medicinal properties. So absolutely, these kinds of things do have value. One more, the scientific method, sleeping pills. I'm going to read this one to you as well. Daniel Kripke is a researcher at the University of California, San Diego. He has no relationship with any drug company, so what that means is he is theoretically free from bias, or at least more free than somebody who is, say, paid for by some kind of drug company. In 2012, Kripke examined health outcomes for people taking sleeping pills. He examined 10,000 people taking pills, most of whom were taking Ambien or Restoril. By the way, this is a real study. And it comes from a book called Why We Sleep. Great book if you've got a lot of free time. He compared them with 10,000 well-matched individuals of similar age, race, gender, and background who were not taking sleeping pills and controlled for body mass index, history of exercise, smoking, and drinking. So all those were controlled variables, and he also randomly assigned people into the treatment group and experimental group. The dependent variable here was mortality. That means how many people died across a 2.5-year window. The next slide is going to be the results. The results. What conclusions can be made? On the top of this column, it is group or number of pills taken per year. So in each of these groups, there was more and more sleeping pills taken per year, with the bottom being more than 132 sleeping pills taken per year, middle group being 18 to 132 pills taken per year, half to 18 pills taken per year, and then the control group. The control group is just your average person, and what we're interested in, the dependent variable, is mortality. So the control group is used to as a comparison. Basically, how many people in a random population are likely to die every year? Well, X number. I've just used the number X to simplify things. And what we see here is that if we look at a group of people who took between half and 18 pills, sleeping pills, per year, their mortality was 3.6 times higher. 3.6 times higher. And then as we go to increasingly more and more sleeping pills taken, then what that means is the data is pretty clear, more and more mortality. So what conclusions can be made here? It's kind of a difficult one. Firstly, most diseases that, well, we'll just say most diseases. A couple examples would be cancer, let's see, fatty liver, let's see, pneumonia. A huge number of diseases, and close to all of them have some negative effect on sleep. So it is possible that the mortality, the increase in mortality between the average population and taking a small amount of sleeping pills, it's possible that this group of people simply had underlying diseases such as cancer, and that they were therefore seeking out sleeping pills because they already had a life-threatening disease. So we still have to be careful to not jump to conclusions. We can't say that taking sleeping pills actually increases mortality. And why is that? Well, basically because we aren't controlling for the type of person who is going after these pills. The people who want these pills, that's the problem, is that they are desiring to have the pills. And it could be that they are desiring to have all these sleeping pills because they have other diseases going on. So what we would really need to do is take maybe 10,000 people, randomly assign them into two groups, give half of them sleeping pills, and then the other half we don't give any sleeping pills. And the problem with that is that it is unethical. And what this means is that basically every drug is tested on animals in that way to make sure it's safe for people. So we can say that, boy, I really wish that I had this information before I chose to take sleeping pills. But this is not definitive evidence that taking sleeping pills increases mortality. So, hypotheses. Your book talks a little bit about this, so I'm going to talk a little bit about it as well. Hypotheses, they must be testable and falsifiable. So, a hypothesis is basically a prediction about how an independent variable affects a dependent variable. In our last example, the independent variable was taking sleeping pills, and the dependent variable was mortality or frequency of death. So any good hypothesis must be testable and falsifiable. It must be possible to prove the hypothesis wrong. Here are several hypotheses, and we can try to rank them from best to worst. A, B, C, and D. So A, and you could probably come up with several different orders of ranking. So we'll just say good and bad and terrible. A, ladybugs are a good natural pesticide for treating aphid-infected plants. So you want to think, is the independent variable clearly identified here? Yes, it is. And that would be presence of ladybugs. We could have phrased it a bit better, such as plants with ladybugs on them will have fewer aphids. That would be a better way of phrasing it. A good natural pesticide, good is not measurable and it is also subjective. What is good, what is bad? Perhaps you simply hate ladybugs. So this is not a measurable term. What we'd really be looking at if we want to improve this hypothesis would be the more ladybugs there are on a plant, the fewer aphids there are on the plants. So that therefore we can count the independent variable, how many ladybugs, and we can also count the dependent variable, aphids. Measurable. B, when there is less oxygen in the water, rainbow trout suffer more lice. This is a little bit better in that less oxygen, at least we can measure two bodies of water and say this one has less oxygen and this other one has more oxygen. And then we can also measure more and less lice. So this is a little bit better than A. C, natural cures are better than those sold in pharmacies. This has got to be an absolutely terrible hypothesis simply because, number one, natural, what on earth does that mean? It's just simply because it comes from a plant. Does that mean it's natural? A lot of plants have poisons in them. And then also, those sold in pharmacies, many natural cures are likely sold in pharmacies. I'm not an expert on pharmacies. So this is just super vague and a great way to perhaps start an argument because it is so unclear. Hermit crabs like colorful shells. This one also is terrible because like is not something we can measure. You could perhaps measure in human beings because we are able to speak and communicate really well, but hermit crabs are not known for their conversational skills. So C and D, absolutely terrible. And then we'd probably want to go with B as the best, and then A as sort of a middle ground. Simply, and the problem being is good natural pesticide. Good is not measurable. So, major themes in biology. Our book goes through 5 of them, and if you looked at another book, they might say there's 6 themes, and then another one might say there's 4. But the authors later on in this series are going to keep coming back to this. So, first one is biological systems interact. For example, in a single body, you're going to have different cells. This is a neuron or a kind of a brain cell, sort of. It's not actually a brain cell, but it's a neuron, and then it's affecting some kind of other cell. So systems interact. This cell is interacting with this cell. Here we have red blood cells flowing through a blood vessel, and they're carrying some oxygen, and they are going to interact with the surrounding tissues. When they get to a low oxygen environment, they are triggered to release their oxygen. They are interacting with their surroundings, which happens to be part of the living system. Goes also to the level of ecosystems or food webs. Here we have a plant against its will interacting with an insect that is eating the plant, and then another insect, this ladybug, is interacting with this bug by eating it, and then the bird eats that bug. So whole communities of living things, this would be a community, a lot of different species living together, they interact with each other. This is a topic for a later course in the 200 level series, as is red blood cells, as is this one about nerves, but that's the examples that they look at a little bit. Major themes in biology number 2, structure is related to function. Here, this is obviously the bone and jaws of a cow. Obviously, it is not the jaws and bones and teeth of a cow, and you all know this. I was just kind of waiting for you to sort of realize I'm lying to you. These teeth suggest very obviously that this animal is not going around grinding up grass. These canines are for ripping and tearing flesh. Its molars even have been modified to basically rip and tear more flesh. And if you've never seen this happen, well, I'm not going to say go out and try to watch a predator eat some poor hapless animal, but you could maybe watch your dog eat something, and you'll notice they don't really chew much because they don't really have molars. They're more of the ripping and then swallowing as soon as possible type of critter. The owl, also not a chewer, clearly. What is the beak for? It is for biting a smaller animal, and then, yes, it doesn't chew either. It's just going to basically swallow it whole. We could probably predict that the esophagus of an owl is very flexible. Structure is related to function. Third major theme in biology, information transfer. So within an organism, between organisms, and from generation to generation. So getting into a little bit more detail on this one. Within an organism, same thing. Neuron is affecting this cell here, and again, structure is related to function as well. The structure of this neuron is long and thin, and that's for transmitting information. Between organisms, well, I'm not sure if these two owls are exactly communicating with each other, but they are definitely about to transmit their information from one generation to the next generation, and that is specifically in the form of genes. That's the only thing living critters can transmit to the next generation. And they can also make a whole bunch of noise between individuals, and that's definitely information transfer between different organisms. People are pretty good at that. And baby owls are awesome, so let's just look at this baby owl, looking kind of dangerous. Used to be cute with its fluffy outer layer, now it just looks kind of creepy. And one more, just because babies are awesome. Cute owl. Information transfer. Okay, so information in living things, all living things, whether it's a bacteria, a redwood, or a jellyfish, the information that they store is in their DNA. Whoopsie, is in their DNA. We'll come back to this in a later chapter. That information is held in C's, G's, A's, and T's. The alphabet of DNA is very, very small. DNA carries genes. Next, again here, cellular level, information can be transferred from this, nucleus all the way down the length of this structure. It's called an axon until it gets to the next cell. Now let's go back to this idea of information transfer, DNA. What DNA does, the main thing is it produces proteins, and it codes for what proteins are produced. So basically, the difference between, let's say, a Homo sapien and a redwood is that the DNA in a Homo sapien has human genes that produce human proteins and human hormones. This last one here, neurotransmitters, we're going to briefly look at that a little later on. That's what would be happening right here. This cell is going to communicate to this cell by releasing things called neurotransmitters. And some neurotransmitters are, well, like almost like a protein, close enough, sort of. Communicating from cell to cell. What this picture is here is also, this is the topic of an entire chapter we're going to look at. And that is signaling molecules are released inside of organisms, and they will bind to receptors of other cells, and then those other cells are going to transmit that information into themselves, and there'll be some type of cellular response. So information transfer. That's the last one here. Not the last one. Sorry, a little bit more. So the organismal level, this one is just so obvious. Just kind of look at these pictures. See, the dog, it's communicating. It's happy, or at least it's not threatening. That dog, it is threatening. It's angry. This dog, I'm not sure. It's you just turned in your homework, and your dog happens to be a professor, and the dog is not impressed. I'm not sure. This one, information transfer. Well, sort of information transfer. It's actually, I'm going to admit to you, I meant to delete this one, and I should have deleted that one. That's just sort of a weird example. Human beings communicating with each other, information transfer. Are we on the last one yet? I can't remember. Major themes in biology. Again, every book is different. So chapter 1, it's like the author's little special view on biology. Major theme though, energy input and use. All living things require energy. They have to feed on something, and then they're going to use it. And this is a food web showing where each of these creatures gets its energy. The owl gets its energy by being a predator and eating other animals. Where all energy comes from is the sun, and it is converted to chemical energy in plants and producers by the process called photosynthesis. Plants produce things called sugars and, well, pretty much everything else as well, sugars, fats, and proteins. Then all these other critters have to eat plants in order to get energy. That's why plants are called producers. They produce food. All the other things have to consume food, and they are therefore called consumers. Energy transfer and transformation. This one's straight out of your textbook. We're going to start right from the beginning here. Energy from the sun comes in as radiant energy. So that radiant energy is going to be transformed into chemical energy. So photosynthesis captures that light energy or radiant energy, producing glucose. That's a sugar molecule, the product of photosynthesis. A byproduct of photosynthesis is oxygen, and other critters, such as the wolf, is going to consume a producer or some other type of creature. Isn't that weird that they put the wolf down here as if it's consuming the, what is this, maybe an oak tree? That's just a little bit weird. But anyway, some type of an animal would be consuming the producer, and the animal is going to use the energy that the plant has produced. This process is called cellular respiration, and it happens in plants and animals. And what this does is it releases energy stored in chemical form that the plant produced, and the animals and plants use it to get things done, like maintaining homeostasis, such as the wolf would maintain its body temperature, such as growing and developing, reproducing, movement. And when this happens, energy is lost, and when the form that energy is lost in is basically always heat. It's basically always heat. Respiration produces carbon dioxide, and that is a waste product. It's a waste product for animals, but it is necessary for plants to continue doing photosynthesis. So in this way, matter is in fact recycled and reused. For example, the carbon dioxide, that's right, that here, CO2, is used by the plants, and then they produce oxygen, and then the oxygen is used by the animals to do cellular respiration. Then they produce CO2, and then that's used again by the plants, and it's a cycle. Weird thing that they don't mention here is this. The plants are not producing glucose or chemical energy so that animals can eat them. No plant wants to be eaten. They are producing chemical energy so that they can grow, so that they can maintain homeostasis, which would mean making sure that they have the right amount of water, of nutrients that they absorb in the soil, making sure that they grow tall enough to outcompete other plants for light. But unfortunately for the plant, some of them do get eaten by animals, and then the animals use that energy. So energy is transferred, it is transformed. The types of energy transformation we've talked about just now would be radiant energy, chemical energy, and then we didn't really show it on this diagram, but there would also be heat energy. All right, over here, what we are looking at again is basically the flow of energy photosynthesis, plant gets some energy, yay, and then the caterpillar eats it, and then the bird eats the caterpillar. So the energy is going from sun to plant to herbivore to carnivore to predator. And where does all of this matter go in the end? All the matter is going to basically go with the organism when it dies, and it's not quite finished getting used because their dead bodies still contain some chemical energy, which is used by decomposers, which are mostly bacteria and fungi. A lot of insects and other types of usually invertebrates are also decomposers like worms, but we're going to focus on bacteria and fungi here. What they do is they break down their dead bodies into their constituent nutrients, such as nitrogen, phosphates, and this is going to add those things back to the soil. Energy, on the other hand, is not reused, not recycled. Energy input from the sun is absorbed, turned into chemical energy, and as soon as it's used, it is lost as heat. It's lost as heat, and then the organism will use some of the energy to get something done, and that is also the topic of another chapter, which we'll talk about later. Animal here, also energy lost is also lost as heat, cannot be reused, but all the material in their bodies can be reused because of decomposers. So flow of energy, this is basically more or less what I just said, but it is in term form. So from sunlight, that is radiant energy, to producers, that would be plants, and this would be in chemical form now. Now it's in chemical form. To consumers, that would be, there's a couple kinds of consumers. We're not going to get into the details, but herbivores and carnivores, things that eat stuff. And then there's decomposers. And the decomposers make it possible to recycle a lot of those nutrients. Every time energy is converted from one form to another, sunlight to radiant, sunlight to chemical energy, and then from one chemical form to another chemical form, heat is lost in every type of energy transfer and transformation. So, in conclusion, energy is not recycled, and it can be changed from one form to another. Over here, you can pause and look at these. I'm not going to talk about them in detail, though. Light to chemical energy, electrical to thermal energy when you heat a pot of water, chemical energy and ice cream to mechanical energy, riding around wondering why you ate that whole tub of ice cream if you're somebody like me, and then chemical energy to mechanical energy. We drive our cars by putting chemical energy into the car. Major themes in biology. Last one. Holy smokes, there's quite a few. Populations change over time. So this is about evolution. And before we get going on it, simply because I know that some people might be a little bit uncomfortable with it, so let me just phrase how I think a productive way of thinking about it is. Consider the fact that sometimes in our country, in the United States, we sentence people to death for crimes they have committed. Consider this. Sometimes nobody saw them commit the crime. So what happens is police gather evidence. They submit this evidence to a jury. The jury is committed to understanding the evidence and then deciding, is this person guilty or not guilty? Essentially, and in this crime, nobody saw it committed. Nobody was there to watch it happen. Yet, even without any eyewitnesses, we are sometimes willing to believe in evidence so strongly that we will put people to death. So, with regard to evolution, it is a very small amount, part of this course. Usually, I don't even see it until the end of a book, but they put it in the first chapter in this one. Think about it like a jury thinks about an important murder trial. Your charge is simply to understand the evidence gathered by scientists in this case, and then come to your own verdict. So you're simply required to understand the evidence of it. So here we go. A couple of little bits of background though. It was not discovered, but the theory was put together by Charles Darwin. He basically got on this boat. He was seasick for, was it 3, 4, 5, 6, 7 years? He was seasick like every day, but he really liked what he was doing. He observed a whole bunch of stuff, and he came up with the theory of evolution by natural selection. Your book mentions adaptation specifically. An adaptation is inherited characteristics that enhance an organism's ability to survive in a particular environment. And that is essentially what Darwin observed going around the world. He just kind of noticed that, wherever I go, it seems like creatures tend to be adapted to where they live. For example, birds are generally flying animals, and then this one flies too, but it has webbed feet, and what do you know? This bird swims around in the water a lot, so it's adapted to swimming in the water. Most mammals are land dwelling. There's some exceptions like whales and dolphins, but most are land dwelling. This one is adapted to living in the water a lot. Still got legs to run around. This is the hind foot of a beaver, but notice the webbing in its feet. Here we have a moth, and notice the moth is just really well camouflaged. This is an adaptation. All of these are adaptations. So, evolution. Definition is the process by which populations of organisms change over time. And that is, I basically just copied that exactly out of your textbook. Just another note on things to pay attention to in your textbook as you're reading. Words that are put in bold or darker or black writing, you want to make sure that you get all of those down. And evolution was one of those words. Theory is another one, and theory, I kind of took this one out of the scientific method section of your textbook. Also copied this one because every book has a different definition of a theory. They're all similar, but they're all a little bit different. So your book says integrated explanation of some aspect of the natural world that is based on a number of hypotheses, each supported by consistent results from many observations and experiments. And that is essentially what evolution is. A whole bunch of experiments that tested various types of change over time. For example, we're going to go into one example, and there have been so many experiments, and all together they support this theory of evolution, a process by which populations change over time. So we're now going to look at one such experiment. Well, it's not so much of an experiment because it was looking back in time. Again, the jury analogy is appropriate because a lot of what evolution is we didn't see it. Nobody was there to see it happen. Some hypotheses can't be tested. Right, and that is to some extent that is true in evolution. For example, we can't go back in time and test if some extinct fossil will eventually evolve to become reptiles and mammals. You can't really test that hypothesis. How can it sort of be tested? Well, you can say that if somebody is suggesting that birds, reptiles, and mammals share a common ancestor, they would say, therefore, you would kind of expect to eventually dig up a fossil that is sort of a mix between all of them. And then, well, basically, they did find a fossil that's kind of a mix between all of them. And then you might say, still, again, well, if this fossil led to birds, to reptiles, and then to mammals, we would expect to find more intermediate forms in the fossil record. And they've also found more intermediate forms, and we continue to find more and more of them. So yeah, some hypotheses cannot exactly be tested, but we can look back in time to some extent.
Here is a story of
industrial melanism. So industrial melanism. Basically, what we have here is there's two types of moth that we can see here. Let me get the writing here. And the species name is Bistombecularia, the peppered moth, and it is active at night, flying around at night, and then it rests on trees during the day. So to avoid predators during the day, it relies on camouflage. It doesn't run away at all. It basically just lands on a tree and hopes no predators see it. All insect collections prior to 1849 had pale wings, so like this, like, like that one, pale wings with black specks, but in 1849 a dark-winged moth was found. Over the 19th century, the black or melanic form of the moth increased in frequency in some areas and decreased in frequency in other areas. So this experiment, by the way, was not done in real time. It was done by looking into the past. Some people, myself included, find it to be very enjoyable to collect insects, like these beautiful butterflies. I tried to find a picture of a moth collection, but well, moths are a close relative of butterflies, and every time you put in any word for a moth collection, butterflies come up. So anyway, they basically looked at moth collections. They'd be something looking like this back in time, and all insect collections, they're basically labeled in a standardized way, such as where was it found, when was it found, and what species do you think it is? So we're able to look back in time at the abundance of these speckled or white moths and these black moths. So, and what they found was that over the 19th century, the 1800s, the black form of the moth increased in frequency in some areas, and then also decreased in other areas. So what's the deal here? What's going on? Turns out this is the deal. So, over the 19th century, here is the United Kingdom. Well, here this is Ireland. Don't want to start any big arguments if any of you are Irish. Sorry. So Ireland, and then the mainland, England and Scotland up here. Industrial revolution occurred, and they put so much smoke into the atmosphere that the trees were darkened, and also this kind of white stuff here, it's a kind of lichen. The lichen was also killed, and so the trees became darker colored. The trees became darker colored. You can see this tree darker colored. And what that did was it favored a different phenotype or visible characteristic, visible trait. The darker moth in polluted England now has an advantage over the lighter moth. So which moth do you expect to have more offspring? Each one will have between maybe 40 and 1,000 offspring. The black moth is much less likely to get picked off by predatory birds. The white moth, it's very visible, is more likely to be picked off by predatory birds. So over time, the population of moths, Bistombecularia, changed, becoming a darker color. So, in conclusion, let me come back to these 4 terms that are general terms about evolution. I think they'll make a little more sense now that you've seen this example. You need to be aware of these 4 ideas. So number 1, natural selection is these 4 things, and natural selection is the mechanism by which populations change. Firstly, Darwin observed that there is variation in all species. So even prior to the 19th century, there was some variation. Most of the moths were the white speckled form. A couple were black. Very few were black, though. And all species tend to overproduce, produce too many offspring, more offspring than the environment can feed or sustain. So most moths do not make it to adulthood and reproduce. Those that are best adapted make it to adulthood and leave offspring, passing on their genetic material to their offspring. And this is known as competition. And not all individuals compete with other individuals by fighting. It may simply be they're competing by who is the best camouflage. So in polluted England, the darker moths were more successful competitors. They were more adapted individuals, and they had more offspring, and their offspring tended to look like them. In conclusion, Turns out that once, I think it was the 70s, they passed the Clean Air Act, scrubbed England's skies clean, the lichens came back, and turns out that the moths went back or reverted to being speckled colored moths eventually in the end. Reverted, changed back to being speckled colored for all the same reasons. Here we have the 6 characteristics of living things, and you can see them down below. 1, they're composed of cells. Everything is made of cells. 2, they grow and develop. 3, all living things evolve and adapt, have a self-regulated metabolism, respond to stimuli, and reproduce. And this slide is a visual representation of the 6 characteristics of living things. This one kind of represents the fact that all living things evolve and adapt. Its feet still have the remainders of a 5-fingered limb in there, if you look at the bone structure, but it is highly adapted to swimming because it has essentially, well, not fins, but flippers. All living things are composed of cells. If you look at an animal under the microscope, well, it's not that interesting. They pretty much all will look like this in some part of their tissues. Actually, I should say it is interesting, but when you look at them all and they look the same, it's a little less interesting. All living things grow and develop. A little baby crocodile. Responding to stimuli. This could be a good one. A good example of responding to stimuli. Have self-regulated metabolism. This is a Venus flytrap responding to stimuli. The insect has triggered it to close its trap. It's also regulating its metabolism. The reason these eat insects is to get nitrogen. They typically dwell in really nutrient-poor soil in the tropics. And then a baby penguin. All living things reproduce. Next topic is taxonomy and classification. It is hierarchical. What does that mean? It means that we go from very general, such as is it alive, to very specific, such as what species is it? Is it a dog or is it a cow? And then everything in between is, going up. We're getting more and more broad. For example, kingdom. The kingdom, you can maybe remember that from high school. There's 4 of them, plants and animals and fungi or protozoans. So those are very general, and then species is very specific. First, the 3 main domains in the phylogenetic tree of life. So domain is right up here, even more general than kingdoms. At the domain level, we are basically looking at characteristics of cells. Bacteria, archaea, and eukaryota. Those are the 3 domains. Let's start with the easiest one, eukaryota. Those are all organisms that have cells that have a nucleus and other organelles, such as the mitochondria, a chloroplast, and a whole variety of other things. Bacteria and archaeans used to be in the kingdom bacteria, but then we decided to change things up a little bit and create these domains. So bacteria are thought to be sort of the oldest, and they are also the simplest of creatures. Bacteria are prokaryotes, meaning they have no nucleus. Archaeans are also prokaryotes. They have no nucleus. What is the difference between bacteria and archaeans? We're not going to get into it today, but suffice to say, archaeans are bacteria that live in very extreme environments. They are also called extremophiles. For example, one group of archaeans here are called halophiles. That means they live in extremely salty environments. This one down here, Pyrodicticum. I'm not familiar with that one specifically. Pyro, though, I bet you anything, it lives in a really hot place. Thermoproteus definitely lives in a hot place. These things have been highly studied in the hot springs of Yellowstone. Methanococcus, that is definitely going to be a single-celled archaean. They're all single-celled, but a single-celled archaean that lives in an environment rich in methane, and then these ones are also the same thing, Methanobacterium, Methanosarcina, etc. So, no nucleus, yes, has a nucleus. At the kingdom level, we have plants, fungi, protists and animals. Let's talk about the first one first, the easiest one first. Animals, that's us. What are we like? We have lots of different tissues. We are pretty much always multicellular. Let's see. We reproduce sexually, and we have a lot of different tissue types. Plants, they are kind of similar to us. They are also pretty much always multicellular. They have many different tissues, maybe not quite as many as animals. They are producers. They can do photosynthesis. They can reproduce sexually and asexually. And they have chloroplasts. So they are the only kingdom where chloroplasts are present. Fungi, they do not do photosynthesis. They do not produce their own food. Fungi are decomposers. They break down dead organic matter and then absorb the resulting nutrients. Protists. This is the most difficult one to remember. You can also call them protozoans, and they are always, pretty much always, there's always exceptions in biology, variation. They're pretty much always, though, single-celled, and they are also eukaryotic. They are, so notice here, eukaryotic. So they have all the same cellular structures generally as animals, fungi, and plants, such as a nucleus. We'll talk about this later in the next chapter, but they also have a, what else do they have? They have a nucleus, a rough ER, a smooth ER, a mitochondria. Some of them might even have chloroplasts, which makes it really confusing as to what exactly are they. Some of them are very plant-like. They could have a chloroplast. And then others will have a flagellum looking sort of animal-like, but they are all single-celled eukaryotes. And then down here, we have the prokaryotes, no nucleus. How do you remember all of these different classifications? Holy smokes, there's a lot of them. Well, the way I like to do it is come up with a mnemonic device or a memory aid. And I'm sorry if this is spelled incorrectly. I used to teach British curriculum, so sometimes I accidentally spell things in the British tradition. Anyway, D, K, P, C, O, same thing, same first letter as these, right? Domain, kingdom, phylum, class, order, family, genus, species, and then subspecies down there. The mnemonic device I use is as follows. Does Canard play classical or folk guitar songs? Does Canard play classical or folk guitar songs? There we go. Gotta memorize them. I'm sorry, you do. Again, here, most general. Here, most specific. Then in the middle, we could have things like class mammalia. And then here we have something such as kingdom animalia. And then as you get closer to the species level, you get more and more specific, such as Homo sapiens. The binomial naming system, this is how we name a species. For example, human, what is that in another language? The only other language I can say it in, I think I might mess it up, I believe in Spanish it's humano, Ningen in Japanese, and I am terrible with languages. So the point being is it gets very confusing because science is an international endeavor, and if we have a different name for every living thing in every different language, it becomes very difficult to communicate with each other about what we are studying. So the binomial naming system is very specific. Binomial nomenclature. That means the two-name naming system. Homo sapiens. Homo is our genus, and it is always capitalized. The genus is always capitalized. Sapiens is our species name, never capitalized. If it has been used once in your piece of work or your test answer, for example, the name can be abbreviated to the first letter of the genus, that's the genus up here, and then the full word for the species, or H. sapiens. So Homo sapiens, the genus and the species. If we are typing, then we are going to always italicize the binomial name. So Homo sapiens here, you can see it is italicized. If it is handwritten, it must be underlined. So we are Homo sapiens. Looking at this slide, which of these two are most closely related? This is a little bit of a tricky one because there is a subspecies in here. Canis lupus familiaris, or doggy. Canis lupus laniger, the Tibetan wolf, and Canis rufus, the red wolf. Which two are most closely related? You can pause for extra excitement or just go with me to the next part of it. These two fellas here are most closely related because they are the same species. There's the genus, there's the species name. Here's the genus, here's the species name, Canis lupus. They are different subspecies, Laniger and Familiaris. So they are, in fact, yep, the same species. And another way that we define a species is, can they interbreed? These two fellas can interbreed. This one over here, Canis rufus, or the red wolf, this is in the same genus, but it is a different species. Now, unfortunately, I got to tell you, it's a little bit of a sloppy definition. Sometimes members of the same genus, so Canis, can still interbreed, even if they are a member of a different species. For example, zebras and horses can interbreed. Donkeys and horses can interbreed. Lions and tigers can interbreed. Polar bears and brown bears can also interbreed. The list goes on. Which of the following are true of these elephants? 2 and 3 are. They are two species in different genii. Two species in different genii. Loxodonta africana and Elephas maximus. And number 3, they are from two genii in the same family. So this was kind of not really listed here, but yeah, all elephants do belong to the same family. And so if you'd like, you can discuss these questions, test your memory, list 5 major themes of biology, list 6 characteristics of living things, what are the 3 domains of living things, and can you describe evolution by natural selection?